Welcome to BioRatna. Dear students, I am Chandana Sengupta and our today's topic of discussion is HGP. That is Human Genome Project. One more interesting topic from the chapter Molecular Basis of Inheritance. Let us see what is genome and what is a genome project. So genome is the complete genetic information. It is the complete genetic information which is present in the haploid set of an organism. So what is genome? It is the complete genetic information. Complete genetic information present in the haploid set of chromosome. So in human beings, what is the diploid number of chromosomes? The diploid number of chromosomes, it is 23 pairs. So the haploid number of chromosomes, it is 23. So whatever information is present, genetic information present in these 23 chromosomes is called genome. Now let us see what is Human Genome Project. It is a very ambitious project, students, to find out the complete nitrogen base sequence. You know that on the DNA the nitrogen bases are arranged one by one. The nitrogen base pairs are there. So to find out the complete nitrogen base sequence in the haploid set of chromosome is Human Genome Project. So it is to find out or to sequence the complete nitrogen base sequence in the haploid set. Because the other set, it is the homologous chromosomes, so it is having the same information. So to find out one's information in one set is sufficient. So this is genome and this is the Human Genome Project. Just see the journey of human beings, students, struggling to find out the structure of DNA. Now the stage has come where they have taken this ambitious project to find out the complete nitrogen base sequence in the haploid set of chromosomes. The chain of thoughts which led to the quest to find out this nitrogen base sequence is that the characters of every individual is decided by the nitrogen base sequence present in his or her DNA. So if two individuals, they are different in any character, that means there is difference in the sequence of nitrogen bases in them. So if we are able to find out this difference in the nitrogen base sequence of them, then this is going to revolutionize the diagnosis, treatment, and maybe someday we can prevent these diseases. Thus started the ambitious project of HGP. Let us have an idea of the total number of nitrogen base pairs which is present in the haploid set. If it is in diploid set, then the number is 6.6 into 10 raised to the power 9 base pairs. This you have studied while studying the packaging of DNA. So in haploid set, this number is 3.3 into 10 raised to the power 9 base pair. So you can see what a vast work it was. It started in 1990. It started in 1990 and it was 13-year project. It was 13-year project to be completed in 2003. The sequencing of 22 chromosomes was completed in this time, but only the chromosome number 1, its sequencing was completed in 2006. Only chromosome number 1. Otherwise, all others, it was started in 1990 and completed in 2003, that is in 13 years. You should know, students, that chromosome number 1, it is the longest chromosome of all these 23 chromosomes. You should also know that while doing the karyotyping, the chromosomes, they are arranged according to their length. So the chromosome number 1, which is the longest, is the first one. That's why called chromosome number 1. Then the second one is little bit shorter than it. Third one again a bit shorter, and so on. HGP was a mega project. Why mega? Mega means bada. Mega project. Why is it called mega project? We will see two reasons for that. First, the cost. The cost of sequencing the nitrogen bases was huge. You know the number of nitrogen base pairs present now? It is 3.3 into 10 raised to the power 9 base pair. And the cost for sequencing of one base pair was US $3 per base pair. Per base pair, let me remove it from here. It is $3 per base pair. That means to sequence all of these, you need $9 billion. $9 billion. This is the reason why HGP is called a mega project. Now, let us come to the second point. Why it is called mega project? The information which was generated, students, it was huge. Large amount of information. So huge that your NCERT tells you that if it is in books, naturally it was not possible to print in the book and handle it. It was all stored in the computers. But to make you understand how huge amount of information was generated, so your NCERT tells you that if the information is in book form and one book is having 1000 pages and each page is having 1000 letters, then how many books will be required? 3,300 such books will be required. That huge amount of information was generated. That's why naturally it is a mega project. Actually, it gave rise to a new branch of biology called bioinformatics, so that this huge information can be stored properly, can be analyzed properly, can be handled properly, and the data can be retrieved from it in easy way. Bioinformatics. Now let us see the goals of HGP. The goals of HGP. You will say that we already know it. Why do we need to learn it again? We know that the goal of HGP is to find out the sequence of all nitrogen bases present in the DNA. But if you find out the nitrogen base sequence and you are not knowing that how many nitrogen bases are forming one gene, what our aim is, not just to find out the sequence, we want to apply it. Our intention is that if one person is having thalassemia, one is suffering from thalassemia and other is not, then what is the defect in the gene of the person who is suffering from thalassemia, which nitrogen base differs from the healthy one? So we need to identify the genes. So our first goal is to identify. We have approximate 20,000 to 25,000 genes. So to identify the 20,000 to 25,000 genes present in human DNA. This is first. Second one you already know. To sequence the nitrogen bases in DNA. कर लिया, सीक्वेंस कर लिया, काम खत्म हुआ? नहीं, असली काम तो बाद में शुरू होता है. When you have found out the complete nitrogen base sequence, now you have to save it properly. You have to analyze it properly. So the next point is to save the data, to save the information in database. Store कर दिया. Now to improve tools for data analysis. You can see that without these things, just finding out the sequence makes no sense. You have to store the information properly in the database. You have to improve your tools for data analysis, and then the purpose was not just to store it, but to transfer the information to different sectors. So to transfer the information to different sectors for which it will be useful. Now the last point, that is ELSI. What is ELSI? You can say that it is byproduct. No, you can't say it byproduct. It is side effect. Side effect of HGP means what human beings are trying to do, means the scientists are trying to do, that if one person is suffering from thalassemia, one person is suffering from any disease and the other person is healthy, then what is the problem in the gene, in the nitrogen base sequence of the person who is suffering, which is making him to suffer? To find this out, so that to find out the cure for this also. But many people, they think that this is against the law of nature. So many issues, this I is for issues, na? So many issues they arose, and so to deal with all these issues, this is, what are these issues? They are, E is for ethical, L is for legal, S is for social, and this I is for issue. So to address, so this is to address the ethical, legal and social issues which arose due to HGP. Now we should know that which countries participated in this huge mega project. How to remember the name of the countries? When you think of the developed European countries, then you talk about UK, Germany, France, and where will US go? US has to be there, US, and from the east side it is China and Japan. Actually, the work was started by US. Two departments of US, they took active participation and they started this project. These two institutes, they are Department of Energy and National Institute of Health. These two departments, that is the US Department of Energy and National Institute of Health, they started it. And from UK, the Wellcome Trust, it became a major partner. Thus we have learned this was the first part of Human Genome Project. In this first part of HGP, we have seen what is HGP, what are its goals, which countries participated in it, why it is called mega project, etc, etc, etc. हमारे सेकेंड पार्ट में हम देखेंगे कि हमारी इस धुन ने, हमारी इस लगन ने हमारे हाथ में कौन सा खजाना लगा दिया. Other than that, we will be seeing the different methodologies by which we achieved it. I hope this video was helpful to you. Stay tuned, bacha. Stay connected. Do subscribe and thank you for watching the video. See you in tomorrow's Human Genome Project Part 2.